Good evening. Tonight's story comes from Harvard University in Cambridge, Massachusetts. I'm Maya Barry-Ochoa.
And I'm Chloe Whelan, coming to you with tonight's story.
90-year-old Harvard professor Mathew Meselson, 3 days before Christmas, calls the University Health Service to inquire about being vaccinated against COVID-19.
Meselson was very eager to get his shot, as he has been cooped up in his apartment alone since COVID started. He officially retired from teaching at the beginning of 2020, but continues to study in the lab while wearing a mask, trying to stay safe.
For those who don't know, Mathew Meselson is an American molecular biologist. He was born in Denver, Colorado, May 24, 1930. Meselson studied chemistry at the University of Chicago and did his graduate work at California Institute of Technology. In 1954, Mathew went to Woods Hole to become a teaching assistant. There, he met Franklin Stahl, who later became his partner in their experiment. He worked closely beside Stahl to try and prove DNA replication was semi-conservative. Here's a clip of Stahl and Meselson talking about their experiment.
The prediction of the Watson and Crick model is the two parent chains come apart, each one makes a new daughter molecule, and that's replication. So that would predict that after exactly one generation, when everything has doubled in the bacterial culture, that you'd find the DNA molecules all have one old strand, which is labeled heavy, and one new strand, which is labeled light, and therefore their density should be halfway between fully heavy and fully light. That would be the prediction for what you see at exactly one generation.
What do you predict to see for the next generation? Well, each molecule would again separate its chains, one of which is heavy, the other of which is light, and the only growth medium available is light growth medium. Then the light chain would make another light chain to go with it, a complement. The heavy chain would make a light chain to go with it. So after 2 generations, you have DNA, half of which is half heavy, and the other half of which is all light. And fantastically, that's exactly the result that one could see.
While working together, Stahl and Meselson worked a lot with DNA and RNA. Their goal was to either prove or disprove Watson and Crick's model of conservation replication. They did this by inventing a new technique called density gradient centrifugation. This technique is used to isolate and purify biomolecules and cell structures.
Now to Joey for the weather.
Thanks, Maya. This is Joey with the news. Tonight's weather is 55 degrees with clear skies, but it's a little bit dark out tonight, folks. As you can see, Bishop Kelly shut down for the night. Hope all you kids have a good weekend. Back to you, Maya.
Thanks for that, Joey. Now back to tonight's story. While experimenting, Stahl and Meselson proved that DNA was semi-conservative. This means that DNA separates and then makes a copy of itself. They also obtained the data to prove the existence of mRNA, or messenger ribonucleic acid. mRNA is necessary for the production of protein.
In cells, mRNA uses the information in genes to create a blueprint for making proteins. mRNA is used in vaccines because it teaches our cells how to make a protein that triggers an immune response in our bodies. This discovery has impacted today's modern medicine in big ways.
For example, the COVID vaccine contains mRNA, which has contributed to the rapid development of the vaccines. Meselson has won many awards for this discovery, including the MacArthur Fellowship in 1984, the Guggenheim Fellowship in 1966, and the Lasker-Koechlin Special Achievement in 2004.
Thank you for watching tonight's show.
Tune in next time.